Great. Thank you, Frank, for that introduction. And a great introduction, too, because that paper is one of the things I'll be presenting along with some future work or additional work that I've done. So thank you all for coming. This is a great opportunity for me to talk about some of the work that I'm doing. I'm going to be talking about two projects, both looking at factors that shape the dynamics of microbial populations and the biogeochemical processes that they mediate, focusing largely on these aquatic dead zones or oxygen-free zones. And so the first will be a bottom-up approach, looking at the biogeochemical factors, the energy that's available for these organisms, and trying to model them in order to understand where energy is available for different organisms to be found within the water column. And then presenting work about a top-down approach, looking at developing a method to identify host-virus interactions in the environment so that we can understand how viruses regulate microbial communities. So just to begin, I like to start off with how important microorganisms are to all the ecosystems that they inhabit. And so microorganisms are a dominant form of biomass on the planet. And so what they do really impacts the ecosystems they inhabit. So you can see here, this is a 1 microliter drop of water on a teaspoon for your reference. And so in 1 microliter of seawater or natural water, there are about 100 to 1,000 bacterial cells on average. And so you can imagine if you extrapolate that volume to the entire ocean, that's about 10 to the 29th total microbial cells just within the ocean alone. And so all that biomass really impacts not only the ocean and the aquatic ecosystem, but also globally can have very important impacts as well. And my focus is on the impacts that microorganisms have to water and water quality. And so one of the major issues that we face in terms of microbial biomass is algal blooms. So algae responding to a lot of the nutrient inputs from agricultural runoff or urban runoff in urban areas can cause the water to be discolored through their growth. So this can discolor the water, can also create scums on the surface of the water, and potentially create toxins. So some of these organisms create toxic substances that can then cause beach closures and the loss of these resources for people to use. And also, on the flip side of that, all that biomass, then once it starts to be degraded, can fuel additional bacterial processes that use up all the oxygen within the water. And of course, macroorganisms like fish, crabs, oysters, they need oxygen to survive. And so this can result in expanses of, here I'm showing you the Chesapeake Bay, where there are very low oxygen or very little oxygen available for those macroorganisms. And so this can result in a loss of habitat for these organisms and can cause fisheries to be less productive. And so it has a very large economic impact as well. And these problems of oxygen depletion are a major issue, not only within the US, but globally. Oxygen depletion is a major cause of water impairment in the US. So thousands of miles of estuaries, tens of thousands of miles of streams, and millions of acres of lakes are impacted by low oxygen concentrations. And the projections through climate change suggest that this problem is going to be getting worse, and there will be even more volumes and bodies of water that are impacted by low oxygen concentrations. And additionally, along with the loss of habitat, there are microbial processes that are initiated with the onset of oxygen depletion that really have major impacts both to the local ecosystem and globally to if these products get into the atmosphere. And a couple of these things I will point out, especially within the model ecosystem that I'll be talking about, the Upper Mystic Lake. There's actually arsenic within the sediments of the Mystic Lake, which has been washed down from a Superfund site, which is upstream. And so this arsenic in the sediment is actually fairly innocuous. There's lots of people who use Mystic Lake recreationally, but as long as the arsenic is in the sediment at the very bottom of the lake, it's not quite as bad and they don't get exposed to it. But microorganisms, once oxygen has been depleted, start to change the mobility of things like arsenic, and this can mobilize it into the water column and move it up closer to where people are using the water recreationally, and so this can be a potential hazard. And we can all, the processes that occur after oxygen has been depleted can change nutrient cycling, either also changing the mobility of things like phosphate as well, which is actually acts similarly to the arsenic within the water column, or also denitrification, which is a process which transforms some of the macronutrients and like nitrate that microorganisms and other aquatic organisms need for growth into a form that's not as accessible to them. And so this can really have an impact on the whole ecosystem food web. And finally, it can produce greenhouse gases like methane, which can be released into the atmosphere and then potentially impact global climate as well. So all of these processes and all of these microbial inputs are very important for us to understand. So that's the goal of my work, is to try to understand and to put these into context, into a modeling framework. Another very important aspect of this is that all of these biogeochemical processes are occurring are very interconnected. And so changing one cycle, like the nitrogen cycle, actually can impact other cycles, like the carbon cycle or cycling phosphorus as well. And so in the particular example of this lake that I'll be talking about, as I said, there's a lot of arsenic at the bottom of this lake. And in fact, arsenic speciation and mobilization is determined by the amount of nitrate in the water column, because nitrate oxidizes iron. So we have the connection between the iron cycle, the nitrogen cycle, and this impacting both arsenic and phosphorus through the microorganisms that are mediating it. So there are all these interconnections that we have to be aware of in order to understand how changes to one part of this food web is going to impact the rest of it. And these changes might not be completely understood unless we can see it through the lens of the microorganisms and what they can do. And so that's the goal of my work, is to understand the microorganisms, their capabilities, and how they impact the ecosystems. And so within the framework of understanding microbial ecology, I think some of the major challenges are to develop a mechanistic understanding of these processes, to link this community structure, who is there, to the functions that we care about in the environment that I just laid out. And one of the tools that I think we can take advantage of is using a lot of the rich metagenomic data that we can get through sequencing which organisms are present, and use that in a modeling framework so that we can bring that information into context, so that we can use it in a predictive fashion. And so we can predict how the microbial community will respond to remediation efforts. Say if we start to clean up around the Chesapeake Bay, how will the microorganisms respond, and how quickly will the bay be cleaned up in terms of trying to get back to a state where there's oxygen at the bottom of the bay. And so that's what I'm trying to work towards, to connect the observations that we get from metagenomics to a predictive framework. And I'll show you some of the work that I've done there. And also, too, I think in terms of prediction, some of the interactions that microorganisms are involved with will be the biggest wild card within this. So trying to understand how we can model and understand these interactions within this context will be important as well. And so I'm going to talk about my research approach, which begins with going out into the environment, taking samples and measuring both the microbial community and the chemical environment that they're found in, and then trying to get that data into a framework so that we can understand some of the factors that control where and when these organisms are found, and how that impacts those biogeochemical processes that we're interested in, like arsenic mobilization or methane production. and ultimately using it in a context where we can understand how the community is going to respond under environmental change scenarios. And so I'll be talking first about this work that I did at Upper Mystic Lake, which is really a model ecosystem for studying microbial ecology. It's used locally by the community there. There are 3 boating clubs and a beach along the shores of the Mystic Lake, Upper Mystic Lake. And the nice thing about this from a microbial ecology standpoint is that it's thermally stratified, seasonally thermally stratified. So basically, in the summertime, the warm temperatures and the sunlight warms the air on the top of the lake and isolates it largely from the cooler, denser water at the bottom through a barrier to mixing at the thermocline here. So basically, what's happening at the bottom of the lake is as close as you can get to putting a sealed test tube out in the environment and watching the microorganisms kind of work to change the environment, because the atmosphere or influences from the atmosphere are reduced under these conditions. And so what you do tend to see then is these gradients in electron acceptors, including the oxygen, and as the organisms used up all the oxygen, they transition to other alternative electron acceptors. So microorganisms can use nitrate or iron or sulfate instead of oxygen. And so they also use those electron acceptors up as they're degrading this carbon that's coming in from the surface. And this creates these gradients, which microorganisms can then position themselves within the water column to take advantage of the conditions that are right for their growth. And so you can imagine them then finding the spot within the water column where they can grow. And so another nice thing about this is that because it's seasonally stratified, every year this process is reset. And so it's kind of like natural reproduction or replicate, so that you can go back year after year to see whether or not the same process is occurring and how reproducible it is and whether or not you can capture that with your modeling. And so I won't talk about the year-to-year variation at this site, but I will talk today about some sampling that I did over the course of about 5 months. So the beginning of this onset of stratification towards the summer. For 1 year in 2003. And we went out and we took samples approximately every meter depth from the surface to about the 22 meters in depth throughout the lake. And we were taking chemical measurements with an in situ probe for oxygen, temperature, pH, ORP, and took some samples back to the lab to look at nitrate, iron, and sulfate concentrations. And then we characterized the microbial community within the lake as well by looking at 2 different measures of microbial diversity. So first looking at just kind of population level, who is there? And for this we used a marker gene, the 16S ribosomal RNA marker gene, which is basically a small name tag for each of the species. And we could do this for a larger subset of the sample, so we could do this over the whole entire 5-month period. And then we looked at the genes that were available too using shotgun metagenomics. So that's taking the entire genomic reservoir, all the genes that are available for these organisms, and looking at that for a subset of them just in the last time point, so in August. And we sequenced them through high throughput sequencing on Illumina machines so we can get lots of this data back. So
I have a quick question. When you collected your samples, how representative is this sample of the whole population that you find across the different?
Yeah, so we do collect approximately every meter, and you can see gradual changes in across those samples. So there's a lot of overlap of the community at the surface in 1.5 meters and some overlap there. So there is a transition. So I think there might be smaller.
That represents basically what you see in the whole. What would you see in the whole deepness of your sample? I mean, your lake.
Yeah, so I think each sample is a snapshot of what's happening at each little site. So they're actually, I'll show you some distributions. They can really align, like I said, tightly to just a very small area. But you can see those organisms mixing to other parts of that. So you do see them mixing around. But one of the potential problems is if we're missing some very fine gradients, that would be at the sub-meter level. And so we have some indication that the gradients don't seem to be that strong, but maybe certain parts of the lake would have these tight gradients. But yeah, good question. So we hope it's, we're capturing all the little niches in the environment, and that the community, we've captured most of it. And so now we have all this data. We have chemical data, we have biological data, but we don't really understand why the organisms are found where they are, or who's mediating different processes that we're interested in. And so this is where, trying to put together some models, not only of bacterial populations, how does the sequence data group into meaningful units, and how can those units then help us understand the community and how the whole community is functioning together as a whole. And so that's where some of the analysis work comes in that I'll be talking about. And so to identify these informative units of bacterial diversity, or groups of genes and sequences, we have to start to use some of the organizational principles to help us identify, okay, which sequences should go together in a group and which are separated. And so you can start to think about this in terms of the organization of genetic information within the cell and community. And so it starts off with genes, which are associated with enzymes that transform different molecules, say, for example, in denitrification from nitrite to nitrate, so on to N2. So there are genes that we know that are involved in this process. These genes, of course, are encoded in the genome of some specific organisms. Maybe they don't have all the genes, but maybe there's a subset found together within a genome. And if that genome is duplicated through the process of reproduction, the whole genome is going to be reproduced. And so those genes are going to co-occur because they're found together within a genome. And so we can use that information to our advantage to identify which genes should be together within a genome. And these genomes are found in organisms, and these organisms are found in populations that have slight differences between them, kind of like the differences in the human population in terms of eye color or hair color, which are not significant in terms of what these organisms can do. So there are slight variations within microbial populations as well, and we have to differentiate that from the important information of new genes giving them new functions that allow them to inhabit different niches. And so we can use the co-occurrence of these organisms, or these different sequence variants, to help us say, okay, this is a group that's coherent and working together. And these organisms, they can have partners. They can pair up with organisms that are making waste products that they can use, or vice versa. And so these relationships can also be identified through their co-occurrence. And likewise, if there's competitive interactions, there will likely to be exclusionary, and so excluding one population from co-occurring with the others. And we can use all of this information to our advantage to understand overall what's driving some of this organization within the community. And so that's what some of the analysis that I have done tries to accomplish. So we looked to identify these populations, both from the genetic similarity of some of the sequences that we're analyzing in the 16S ribosomal RNA data, but also looking at their distribution, where they're found. Are they found, sequence variants found together in the water column? Okay, I think those might be in the same population versus variants that might be in different populations will be found in different places, regardless of how similar they are. So I'm not going to go into the details of that process, but we also were able to infer some of the capabilities of these microbial groups, looking at organisms with similar functions, so maybe some that had been cultured already and were genetically related, and so we could identify what they can do based on bioinformatics approaches, and also this potential interaction by their co-occurrence data. So the bioinformatics approaches we used first was a simple similarity search to those cultured organisms, as I said, so taking these population sequence data and just trying to say, okay, is it similar to something that's in culture, and let's assume that's our first hypothesis about what these organisms can do. And then second, we used a experimental approach to validate the association of some of these populations with the functions that we think they can do. So this is a technique that was developed by Sarah Spencer in the Alm Lab at MIT, and she basically she was able to use this for some of the samples that we have. So the idea here is to try to preserve this physical relationship between a species gene, that 16S ribosomal RNA gene that tells you who it is, and the functional genes of interest. In this case, we're focusing on a gene in sulfate reduction, so DSRB, which is a gene involved in that process. And so if it's found together within the same organism in that genome, then we maintain that physical structure within the genome by isolating that cell and extracting DNA within a polyacrylamide bead. So these genes stay together. They don't start to move apart within the liquid that we're using to extract. And then we use little tiny microreactors, so it's very small droplets, in order to do a PCR reaction to fuse physically the 16S gene with the functional gene of interest. And now when we go ahead and sequence that, we can get a readout of which organisms have these genes and which organisms don't. Because if the organism or the species doesn't have that functional gene, that gene isn't amplified in the sample at all. And so we could use this to experimentally validate some of the bioinformatics approaches that we used. And then finally, we had the metagenomic data, where we could reconstruct the genomes of organisms based on metagenomic binning. And so we used some previously created algorithms to do the metagenomic binning and start to assemble, reconstruct these genomes from the metagenomic data. And we used a method called concoct, which also uses that distribution in the water column to identify genes that should be found within the genome together. And so we have the population and we have this metagenomic data, but it is actually very difficult to merge those two data sets because the metagenomic data, unfortunately, does not do a good job of putting together those population marker genes within the reconstructed genomes. So we used the distribution of the 16S genes, which are shown in black here, with the metagenomic data, and also made sure that the phylogenetic signals were similar. And when we did this, we could find many of the 16S sequences that had these co-occurring genome reconstructions. And those largely supported what we thought these organisms could do based on those cultured representatives. So we have multiple lines of evidence to tell us what these organisms are capable of. And now we want to try to put them together into some understanding of how they're functioning together. So we have all these, we understand a lot about the capabilities of these individual organisms, but we know they work together, exchanging waste products and transforming some of the chemistry of the lake, which of course changes some of the chemistry and so feeds back on which organisms then could be present at any given place or time. And so this is a feedback loop between the organisms and their interactions and the environment. So we wanted to try to capture that with a model. And so my colleague Scott Olson, who is also in the Alm Lab while I was there and is now at the School of Public Health at Harvard University, he drew upon a reactive transport model that was found in the Journal of Hydrology and basically altered that to be appropriate for the water column because it was developed for a groundwater aquifer. And so this basically takes primary redox reactions where you take that organic carbon from those algal blooms that we were talking about, and first up you use oxygen, and then the microorganisms transition to nitrate, some of the irons some of the metals like iron, and then sulfate, and finally methanogenesis. So those are the primary redox reactions that can occur. And all of them create reduced species, so like methane or hydrogen sulfide or reduced iron that can be mixed up into the water column and fuel additional microbial processes as well. And so that's another set of these secondary redox reactions, or another set of energy yielding reactions that these microorganisms can be involved in. So you found all of these together within this context of a reactive transport model to explain how the population structure and the chemical structure develops in the lake, and where we expect to find microorganisms that can do these various processes. And so when we calibrated the data to match our observations, so this is the modeled chemical concentrations, and we tried to match it to the observed chemical concentrations, and this is depth in the water column, and over the course of days from the beginning of spring to the middle of the summer. And you can see the red shows you the higher concentrations and blue lower concentrations. So we calibrated it to match the chemical data as well as we could. And from that, we identified then that the model largely lined up with some of the observations that we had for those organisms we knew were involved in the processes. So the model here is in red, and the black is those population distributions that we identified for organisms that were involved in these processes. And yeah, go ahead.
Sorry.
Yeah, sure, no problem.
In your previous experiment and your previous measurement,
Yeah, this one?
I assuming that you correlate this also with changes in temperature, right?
So the temperature, so you can see here I started at 5 degrees. That's that position at, yeah, so that's the position at depth. Actually, that's that thermocline. So the temperature does vary from maybe about 10 degrees here to about 4 degrees here, but largely it's constant throughout this environmental period because of that thermal stratification. So there's not a lot of changes in temperature, as there would be at the surface where it can go from 4 to 28.
So that's the matter that it's a spring to summer, the temperature is more or less.
Yeah, so it's staying pretty much the same over this time period. So yeah, but very good question about temperature, which is not part of this model at all. We're just modeling reactions that are occurring and movement of those chemical species between water parcels. But so the observations for the model largely capture the model results largely capture our observations, which suggest, okay, we have a hypothesis about what factors have promoted where these organisms are found, and that drive where these organisms are found in the environment. And we can also look at the gene level, so not looking at just the populations, but looking at genes involved in these processes for the whole group of organisms from the shotgun metagenomic data. And you can see that for many of the genes that are involved in denitrification, sulfate reduction, iron reduction, methane oxidation, we're also capturing the gene distribution with this model as well. So we have some sense that this model is working to help us understand factors that are driving the distribution of microorganisms in the sample. So that's one snapshot at a time, right at the very end. So let's look over time. How do the dynamics of these organisms change? Because you can imagine maybe competition or interactions between these organisms might create different dynamics between the process and the population observations. And so I'm showing you here at the top, again, the model for different processes, denitrification, iron reduction, sulfate reduction, and methane oxidation. And the light colors here show you where these are expected over time, again, with depth and over the course of the season. And so at the bottom, I'm showing you here 2 organisms that have overlapping capabilities within the water column. And so 2 organisms that have the genes for denitrification or iron reduction. And I've plotted one organism in yellow and the other in blue. And so the overlapping area where they might be competing or found together are in green here. And so you can see, actually, while some of the organisms do have very distinct areas in the environment that they prefer, which might suggest some metabolic flexibility on their part. Qualitatively, you can see there's some agreement of where these processes are found within the water column and the model. So this is helping us to understand some of the processes that might drive, dynamics over time within this group of organisms. And I'm going to point out here this group of organisms that are involved in methane oxidation because we found some interesting differences between their genomes. So even though they do have the methane oxidation gene in their genome, so both this organism in blue here, 53, bin 53, and this bin 23. The bin 23 actually had the full suite of denitrification genes, so an entire pathway to transform nitrate into N2 gas, which we think they could be actually using to oxidize methane. And this has been seen previously and seen to be very important in some lake systems. Anaerobic methane oxidation with denitrification has been shown to be very important. So we think maybe this could be happening in this lake. But in terms of trying to understand where these processes are occurring, you can kind of see there's maybe this exclusionary one is coming up at one point, and then the other one kind of comes up later, and they're not overlapping quite as much in this area where we think methane oxidation would be occurring. And if we calibrate the model to include this process with denitrification coupled to methane oxidation, this is not what we expect to happen. In fact, we just expect them to be slightly vertically separated. And so we have not quite captured the dynamics of the interactions if there is actually anaerobic methane oxidation occurring with nitrate reduction. And so to understand a little bit more about this process, we added this to the model. And by adding this to the biogeochemical model and calibrating it so it matches the chemical distributions, and using the parameters that we got after calibrating the model with the additional process, and comparing with and without that process, you can see substantial changes to some of the chemicals that we expect to find in the lake. And the most obvious and largest change is in oxidized iron. And so there's a big reduction when you add this process of there's a lot less oxidized iron. And that is thought that is likely because there is an iron oxidizer that uses nitrate to oxidize iron. And so if this organism is actually being removed and is competing, or if this organism is competing with the iron oxidizer and methane oxidation actually wins, then you're going to have a lot less oxidized iron because there's not the oxidant that is able to be used. And so we didn't measure oxidized iron, but we did measure reduced iron, which is actually, so there should be more reduced iron. We also looked at nitrate. So our observations are consistent with this idea that maybe this organism is actually involved in anaerobic methane oxidation in the lake and altering these the biogeochemistry. So this is the ratio of these organisms, so the one organism to the other. And so the anaerobic methane oxidizer spikes around June, where we see a dip in the difference between the nitrate from the model and our observations. And additionally, we see a spike in the reduced iron that we observe, and the difference between that and what is we expect from the model at these time points. So this is suggestive. We still need to do a lot more work to nail this down, but we think that this might be an important process. It's been seen previously in other lakes, and so we want to identify whether this organism can do it, and if it does have the full suite of denitrification genes, which would be unique for a methane oxidizer. And so just to summarize this part of it, the co-occurrence can be very useful in terms of identifying populations and those capabilities and some of the interactions that we think are occurring within the lake that are really impacting chemistry. And the alignment of the gene distributions with the model suggests that we have captured a lot of those important factors that are determining where organisms are found in the lake. And you know, we are going to do a little bit more to investigate whether or not these additional biogeochemical factors are really contributing to a substantial change in biogeochemistry that we should include in the model and try to capture the factors that impact their distribution. And so just before I completely leave this topic and go on to the viral diversity and viral experiment, I do want to say that I'm collecting data to do the same sort of thing in the Chesapeake Bay. So that was, you know, our little test case was the upper Mystic Lake. But I would like to be able to use the same methods and to correspond some of the metagenomic data to biogeochemical models of the Chesapeake Bay to help us predict what happens as climate changes or as our remediation efforts to clean up the bay continue. And so we have been working with some modelers who have pre-existing models of the Chesapeake Bay, either ChesROMs or an ICA model. And these models don't include all of the biogeochemical processes that I would like them to have, so we have to work with the modelers to try to get additional processes into these models. But hopefully they'll be useful in terms of identifying our understanding of where these processes should be occurring, and then we can tie that to our observations of the microorganisms there. And so in order to observe the microorganisms, we did the same sort of thing where we went out in the Chesapeake Bay and just sampled at one position with depth, just like we did at the upper Mystic Lake. And so that's the CB 3.3C station, which is right around the Bay Bridge up by Annapolis. So we went out there over the course of a couple years, so from 2015 to 2017, and collected samples there. But one of the important factors is that actually the physics of the bay are very different from the upper Mystic Lake, where ocean water is coming in is actually submerged under the fresh water that's coming out. And so the water that, you know, the ocean water that's fully oxygenated is actually stratified and separated from the atmosphere down here, and then that oxygen is used up as it travels up along within the Chesapeake Bay along the bottom waters. So we realized that would be an important component to be able to capture in these models. And so we started working with the Department of Natural Resources and Maryland's Department of Natural Resources and Old Dominion Water Quality Lab to have them collect samples for us over the course of their natural normal sampling and observational program. And so they were kind enough to provide us with lots of water that we are in the process of analyzing and understanding the microbial community associated with them. And we're still in the process of looking at this data, but one of the things that I was a little worried about is how responsive the microbial community would be to the biogeochemical conditions, and if it would be just too much mixing, and so they're kind of just mixing everywhere and they just have to be flexible. But so this is data over 2015, where this dark area, dark blue, is the dead zone, so the very low oxygen environment in the Chesapeake. And these are the corresponding microbial community taxonomies. And you can see that while the upper and the lower communities in June, when there was more oxygen available, seem very similar, once oxygen is depleted, then the microbial communities at the top and the bottom of the bay really do start to look substantially different. So we do think that the microbial community is going to be responsive to the chemical changes in the environment, and that we're going to be able to understand something about the processes through their sequence data and the community that we find there. And so this work is ongoing, and I hope to develop something soon for the Chesapeake, similar to what we did for the Mystic Lake. Yeah?
Your methane oxidation anaerobic, I saw the reduced iron has gone down. You are really accumulating there, right? And you had a ORP data.
I had what? I'm sorry?
Oxygen, the
ORP, yes, the ORP data, yep.
So what kind of profile did you see? Is it really getting really negative around that area?
There is a transition. So I don't know exactly when I suspect that the anaerobic methane oxidation is occurring, if it does have a change in ORP at that particular point, but it definitely does drop at about 10 meters, which is right around, between 10 and 15 meters, ORP really does drop generally in the water. So I'll have to look into that a little bit more carefully. But
Because if it is between aerobic methane oxidation versus reverse methanogenesis, the rate of fall will be low.
okay, so that might be an indication of the different processes. Yeah, so I haven't looked into that, but that would be great to check out in the data and see. So I have that, and that's a piece of data that I haven't looked into too much. So yeah, thanks for that suggestion. Okay, so I'm going to transition now to the second part of the talk, which I'm going to be talking a little bit about viruses, right? This top-down control about on the microbial community by viruses. And so even though I told you how abundant microorganisms are, viruses are 10 times more abundant than bacteria. And so they outnumber bacteria 10 to 1 in typical water systems. And up to about 50% of bacterial cells are infected by a virus at any given time. So this is really impacting mortality and the food web dynamics within microbial communities. And just tying it to this anoxic zones, these dead zones, actually viral lysis is likely to make up almost 100% of bacterial mortality within oxygen-depleted zones, because the macroorganisms and the grazers that would otherwise, contribute to a lot of mortality in the oxic areas would have a hard time without oxygen, and so there are probably not as many of these predators, and then the viruses then are going to be contributing to much more of the mortality of microorganisms in these dead zones. So understanding dynamics of populations from top-down perspective, the viruses are going to be very important within these zones. And so viruses are also known, of course, to impact food webs in general. And so phytoplankton, which are the primary producers in aquatic systems, would typically be consumed by grazers, and that energy and resources would move up into the food chain to the larger and larger organisms. But with a lot of viral predation, that productivity gets shunted then to the heterotrophic bacteria as these organisms are lysed, and that dissolved organic material is made available rather than to the grazers to the heterotrophic bacteria. And so this could really potentially impact overall food web structures within any of the environmental systems that we're interested in. So viruses are important for food web, but also too, understanding exactly the patterns of viral infections might provide some insight into stability mechanisms for the community. So some communities might be stabilized if the host range for these viruses is very broad. And so it's basically killing anything that could potentially start to grow equally. And so that could be a stabilizing force. So if the viral host range is very broad, you can have a stabilizing force. Whereas if it's very specific and these viruses only target specific organisms, this might result in predator-prey dynamics, right? That could start to contribute to a lot of population dynamics that would really cycle these bacterial populations within the communities that they're in. So understanding something about the host range could potentially help us understand the dynamics of these organisms, and that ultimately would contribute to their role in the environment and what they can do. And so current methods that try to look at host virus associations, right, looking at their range in which organisms are being infected, all have some flaws in some ways, and some other ways in which they're very good. So for example, a lot of them, like culture-based assays, they would miss a large portion of the bacterial population, which is not cultivatable. And so your culture-independent approaches, where they don't have to rely on something that can be cultured, would get a larger fraction of the total diversity. And bioinformatics approaches and single-cell genomics approaches can get around this cultivation problem, but you have issues with being low throughput or inferring information from this data that we aren't making the specific connections. And so I'm proposing a really cost-effective and high-throughput method, which I think hits all the boxes and does better than any of the other methods out there. And so this method, similar to the method that I had proposed previously for looking at the genes that organisms have, well, now we just target a viral gene and look at the species that co-occur with this virus. And so again, we're taking advantage of that physical association between the host genes and the viral genes and trapping them when we do the DNA extraction within a polyacrylamide bead so that the structure is maintained. And when we do amplification, make sure that that is specific and only amplify organisms that have these viral, that are being infected by viruses, and we don't get amplification from organisms that aren't infected by viruses. The problem with this, though, unfortunately, is that the viral diversity is not well captured by one primer set. So unlike the bacterial diversity, which we can capture fairly well with targeting ribosomal RNA genes, viral genes don't have a universal marker where you can understand which organisms are present through this one primer set. But there are subsets of viral diversity that we can target with some genes that are fairly well conserved, especially the DNA synthesis and structural genes such as the ribonucleotide reductase and some portal genes. That will capture a large diversity of viruses, but by no means the entire viral diversity quite as well as we can do with 16S analysis. So we've developed some primers to target cyanobacteria or cyanophage and focus on cyanobacterial infections within the Chesapeake Bay. So that's our goal. But the first thing to do, because we're developing the method, is to make sure that we're not getting non-specific interactions. So what we don't want to see is two genes, host genes coming together within the same droplet and, making it look like this one host is being infected by this virus when it's not. So how specific can our reactions under this as we are developing this protocol? So my postdoc, Eric Sikowski, has been working on this. And he basically has spiked, using E. coli as our positive control and as the control samples, we have some samples from the Chesapeake Bay that we've been looking at, which don't have E. coli in them and don't have viruses that infect E. coli. And when you just have E. coli, then all of the fusion products that you get out, all of the 16S genes are all associated with E. coli. And when you don't add any E. coli, it's very little, basically at the limit of detection, where we can identify this E. coli gene product. But as he spikes in E. coli cells at different concentrations, you can see that there is some increase, which suggests that's non-specific interactions that are occurring. And so we don't want to see this. We want to see this at zero or basically at our detection level. But you can tell that this is what it was expected if we had got completely random, non-specific binding and fusion of these products. We'd expect the percentage of E. coli to just increase in the resulting sequence data according to how much we put in. And so, of course, we see it's dampened, but it's still not at the level where we can be confident that we're getting very specific interactions, even though this is a very high level of DNA. Even at 30%, this would be about the most concentrated that we would expect to see populations within our sample. So we have some specificity, but it's not as good as we would like, and we're working to develop methods that improve the specificity, including diluting the cells down further to make sure there's less likelihood of them being put together in the same droplet as we do the reactions, and working on some blocking primers that could potentially prevent amplification when we don't want it to. And we also assess whether or not this is specific or reproducible, and we find that high reproducibility as we have different samples. So these are biological replicates from the same water sample, and we can see this is just a digest of some of those products, and if it was reproducible, we'd see the same digest pattern across them. And so for the most part, we can see that there's a lot of reproducibility. So this is still something that we're working on to develop. But we've applied this to an actual environmental sample from the Chesapeake Bay, which is just outside of Annapolis. And from that, we get some indication of what the data will be like. We get some, so here I'm showing you the bacterial 16S genes associated with the samples that we amplified, and the viral genes here along the horizontal axis. And you can see that some bacterial phyla are infected by lots of different viruses, suggesting that they are under very high viral pressure, and we can start to try to identify some of the factors that maybe make them good targets for that. And we also see a couple of situations where the same virus is infecting multiple different types of cells. And so this is starting to look like we can get that host range information that we're looking for out of this data. And so we're going to continue to develop this method further so that we can improve the specificity. But so far it looks like it's going to be a promising technique that we can use to identify these host virus interactions within the environment. And while we have some specificity, we're working on it being completely specific, so we don't have any non-specific products. And the results do suggest that we can get something about the host range out of this in a high throughput manner. So with that, I would like to just thank the people that worked on this. So this work did start with my postdoc and Eric Alm and Scott Olson, along with Sarah Spencer and Harry Heeman at MIT were important in the Mystic Lake work. And then at Johns Hopkins, Keith Aurora Williams, my grad student, worked on some of the metagenomic data, along with Eric Sikowski, who's been doing the virus EPIC PCR and some funding sources. So thank you so much for your attention, and I'll take any questions. Yeah.
It's kind of a basic question, but before, when you were talking about taking oxygen measurements from different depths, right? Do you do it all the same time of day? Because wouldn't they drop a lot at night?
Yeah, right. So Eric, who's looking at the virus diversity in cyanobacteria, has been looking over time, and he's definitely seen a shift, just even in the cyanobacterial population over time. So yeah, of course, there's going to be these shifts. So with the Mystic Lake, we were focusing more on the bottom, underneath about 5 meters to 22 meters. So not as much light is getting down there. There is some suggestion that I think there is a photosynthetic group that might be low light adapted down there, because there is a spike of this one group of cyanobacteria at that lower level. But it's true, there could be some changes over the course of the day that we're not capturing. And I think those changes might be potentially small in terms of the changes across the whole season, where it's really being drawn up from the entire bottom portion of the lake, fairly dramatically. So, but I haven't looked at it. But good question. Yep. Yeah.
In the Chesapeake Bay, when you're in those regions where you have the brackish water, is there much mixing? You made it sound like it's completely stratified, but does the salinity start?
Yeah. No. Yep. Yeah. So the salinity is going to be changing the entire way. So it's going to start as the end members of the fresh and salt, and as they travel up the bay, they get closer and closer together. So the fresh water at the top is much more salty than it was at the beginning, and the salty water is much more fresh at the end when it's still there. But there is a gradient still. You can see maybe the salinity changes from maybe 8 to 20 parts per thousand, from the surface to the bottom where we're sampling. So there still is a gradient, but there is quite a bit of mixing, which was one of the reasons why I was a little bit concerned, the community would just be everywhere and mixed kind of all over the place. But yeah, so salinity does change. Still, there is a gradient.
So is that a parameter in your model then?
Yeah, so we don't have, so salinity is a parameter because it's a very important determinant of the physical environment. So that stratification is going to be largely salinity based, but also there's thermal stratification as well. So it's in the physical models. Whether or not we include it in the biogeochemical models, I haven't thought about including it, but as we start to understand a little bit more about the factors that determine specific communities, that could become a factor. So we could have maybe some processes limited by, these organisms don't survive below some salinity level, and that's actually the factor that determines whether or not this process is occurring. And we'll have to, as we put together this hypothesis and we start to test it, we can see whether that's an important factor. But yeah, good point. Good point. Yeah.
I like the name EPIC PCR.
Yeah, it doesn't. It sounds.
So I have a technical question.
Uh-huh.
So for those, and you at the initial trapping and trying to trap single cells, and then it went through a cell lysis process before the PCR in this very isolated environment.
Yep.
Or is it trapping DNA?
So in the original protocol, which Sarah developed, which has the polyacrylamide beads, yes, you trap the single cell within a droplet, and it's basically just diluted out so that you assume a probability of a single cell, or even less than a single cell per droplet, and then you extract DNA on the beads, but the DNA can't diffuse out of the beads, so it stays within the bead.
So the lysis and all of those things happens, the chemical that
diffuse into the beads, and then
yeah.
but actually now with our latest protocol, we've gotten rid of the polyacrylamide, and we just do a lysis based on temperature. So we just cycle it with PCR, and the cells burst, and that is, we don't even extract or remove any of the proteins or anything like that. We have everything within the cells. So that seems to be working fairly well without any harsh chemicals or any extraction buffers. And it would obviously be difficult to do this on cysts or any really difficult to lyse cells. So I think that's okay for the virus work, because they're probably infecting actively growing cells that might be a little bit easier to lyse. But we haven't investigated it further outside of the cyanobacteria groups, whether or not we're efficiently lysing all the cells with this method. So
the emission PCR in there, when you trap the cells, whether without beads, then it becomes, those beads then get separated into these micro.
So it's all so there is all emulsion based. So we basically just assume a dilution, that we know about how many droplets are going to form and how many cells per droplet, and assume it's all evenly distributed. So we couldn't do this on something that maybe there was a biofilm, or there was some sort of very tightly coupled cells, so some sediments and stuff like that. We would have to develop different techniques in order to separate the cells out first. But yeah, so it's all just dilution based, and there's no sorting, which makes it very versatile, right? So you don't have to have microfluidics capability in order to just use this. You just need a vortex and some, stabilizing reactions for your emulsion, but you could do it in a standard microbiology lab. So that's what we're aiming for. But if we have to, we might revert to the microfluidics approach if we need more specificity.
What's the physical size of these images? Like what in micrometers or
Of the droplets?
Yeah.
I think they're maybe roughly 50 microns or so. So 10 times larger than a cell or so-ish. Maybe it might be 100. I don't know. I mean, so we're not talking necessarily about eukaryotic organisms, right? So we're focusing on cyanobacteria. So yeah. So maybe 50 to 100 microns or so. I'm not sure. But, yeah.
Last questions?
All right, let's thank Dr.
Yeah.
Kugan one more time.
Thank you.
That's great.
Yeah, no, thank you. Thanks for inviting me.
I have a question for you.